between damaging and defensive forces. And this is taken from some pathology book, I forgot which one, listing damaging and defensive forces. We're going to take a look at damaging forces first, and that is obviously gastric acid and pepsin. All right, these are the constituents of gastric juice, okay? You have the hydrochloric acid, okay? Yes, it helps in protein digestion or hydrolysis of the peptide bonds by converting pepsinogen to pepsin. Acid also, we'll cover that in a minute, denatures protein, allowing pepsin to work. But I have here in bold, it impedes bacterial colonization. At present, the general consensus is that hydrochloric acid and pepsin work to really prevent bacterial colonization of the GI tract, okay, rather than digestion of proteins. And that's exemplified by people who have a total gastrectomy. They don't have a problem with nitrogen balance, but they are more prone to enteric infections. Okay. Intrinsic factor is the only indispensable substance in gastric juice, where, as I mentioned, with total gastrectomy, the only thing that has to be replaced really is intrinsic factor so that you have appropriate vitamin B12 absorption in the ileum. Pepsin, as I mentioned, is not essential. Pancreatic proteases can deal with proteins that are ingested, but again, it impedes bacterial colonization. Gastric lipase, that's a minor kind of enzyme, but nonetheless, it's again not important, except perhaps maybe with pancreatic insufficiency or in newborns when lipase is not fully functional in their pancreas. But at any rate, mucus also, you'll see here in bold, impedes bacterial colonization. Okay, so it presents a physical barrier. But along with bicarb, and if you remember in the GI tract, most of your mucus secretion, they refer to it as alkaline mucus. And that's because bicarb is usually present in the mucus to disperse it. We'll come to that. And that serves as a chemical barrier. All right. This is a pretty straightforward slide. It has numbers 1, 2, 3, 4, 5, and they're in the notes defined. We'll go over it very briefly. Carbonic anhydrase, remember this equation is pretty straightforward. It takes the carbon dioxide that's always present in cells due to metabolism and water, and it takes carbon dioxide and water, converts it to carbonic acid, which dissociates into a proton and bicarb. The proton is used by the hydrogen potassium ATPase exchanger, uses ATP as part of its function, and it pumps protons into the lumen of the gland, the gastric gland. Okay, this is done in exchange for potassium. And this channel, potassium channel, is important because it allows for the recycling of potassium here so that you can pump a huge amount of protons into the gastric lumen. Now, by the pumping of protons into the lumen, you have excess bicarb, so the cell has to get rid of it. Whether the cell becomes acidic or alkaline, it's a death knell, if you will. So bicarb is usually pumped to the blood side or interstitium, then enters capillaries, mucosal capillaries, and it is exchanged for chloride. And you'll see in a minute, chloride also contributes to the formation of hydrochloric acid. And this is referred to as the alkaline tide because for every proton pumped into the gastric lumen, a bicarb is pumped into the opposite direction into the blood. And it actually raises the pH of gastric venous blood. Okay, and it's called the alkaline tide. We'll come to the significance of this later. Now, since you're going to need a lot of chloride to join with the proton to form hydrochloric acid, you have this transporter, which transports one sodium, one potassium for two chlorides. So you have two anions and two cations, okay, if you will. And so this provides a huge amount of chloride along with this exchanger to move into the lumen to form hydrochloric acid. The way chloride moves into the lumen is through a channel we refer to here as CFTR, which is defined here as cystic fibrosis transmembrane conductance regulator. It's a mutation in this gene causes cystic fibrosis, and that's another. It's basically you don't die from the GI problems, you die from the lung problems, but it's because mucus becomes so thick in the absence of chloride or some anion to absorb water. Well, I'll cover that in a minute. Basal acid secretion. Now, again, it's a balance between stimulatory pathways and inhibitory pathways. Let's look at the stimulatory pathways first. The most important thing is the gastrin-histamine axis, and that's sort of illustrated here. This is basal secretion, if you will, and it's due to the existence of vagal tone. That's just a constant vagal tone applied to the GI, and here, in this case, the stomach. This over here represents the antrum, and here the fundic region, if you will. Now, the vagus, if you recall, when it comes down, preganglionic fibers come down onto the stomach, they interact with the myenteric plexus and the submucosal plexus. But in any case, you have the enteric nervous system. That's the nervous system within the gastric wall, enteric. Okay, and it releases acetylcholine at the fundus. Acetylcholine will stimulate parietal cell secretion of acid. At the antrum, the myenteric nerves release GRP or gastrin releasing peptide, which obviously impacts on the G cell, which forms gastrin and releases it into the blood. Okay, so this is your neural component. Now here comes gastrin enters the blood. This is the humoral component or the endocrine. Okay, and it impacts on the enterochromaffin-like cell, ECL cell, which makes histamine. Okay, histamine now, this is the paracrine function. It's next to the parietal cells and it activates H2 receptors on the cell and forms acid. Okay, so this is the way acid is being generated by stimulatory pathways. Now, the gastrin-histamine axis I have at the bottom here, as I'm emphasizing it, is the most important stimulatory pathway. Long time ago, they used to think that gastrin worked on the parietal cells, but that's a minor component now. The major component is the gastrin-histamine axis. Look at the inhibitory pathways, and basically there's only one thing you have to know about this, and that's somatostatin. And that's made by D cells. I don't know why they call it D cells, but anyway, they're tonically active as well. Just like the vagal tone, vagus is active. The D cells are chronically or continuously secreting somatostatin. That's the abbreviation for somatostatin. There's your D cell in the antrum, D cell in the fundic region. Okay, so somatostatin is an inhibitory hormone. It doesn't matter almost wherever you run across somatostatin, be it endocrine physiology, reproductive. It doesn't really matter. It's usually inhibitory to other hormones, etc. And here somatostatin inhibits the G cell from making gastrin, thereby interfering with the gastrin-histamine axis. In addition, in the fundic region, these cells directly inhibit the parietal cell as well as the enterochromaffin-like cell. This is a paracrine action of somatostatin, if you will. So this is serving to inhibit. So you have a balance usually between the inhibitory and the stimulatory factors governing acid secretion. And all I'm trying to show you that, let's say you have basally you have acetylcholine coming down, yes, you're stimulating the gastrin-histamine axis. You're also stimulating the D cell or inhibiting the D cells from making as much somatostatin. So the inhibitory effect of somatostatins is minimized, okay? And you're having acid secretion here. In the absence of this, of course, then you knock out acid secretion completely. Conversely, if you have a stimulation of the vagal efferents, and that's either by taste, smell, or vagal reflexes, etc., with food entering the stomach, the vagal efferents increase the firing rate. They're going to increase gastrin releasing peptide here, more gastrin, histamine acid, and they're going to also increase the inhibitor. So that essentially what they're doing is putting their foot on the accelerator in a car and removing the brake. Okay, so that's how you regulate acid secretion in the stomach. So under ordinary circumstances, basal, if you will, basal secretion, you'll have a small amount of gastric juice, if you will, with a low pH. And referred to as this phase, basal, is referred to as being dominated by the SST restraint. And it's, yes, that's one way of classifying it. And again, as I mentioned, with enhanced stimulation, whatever the factors are, you'll be turning off SST, turning on the gastrin histamine axis. I mentioned pepsin as another factor, and yes, every time you have acid secretion, usually you'll have pepsinogen secretion by chief cells, okay? And here is shown acid. The way acid works is, if you recall from your biochemistry, acid denatures protein. Now, it doesn't destroy the protein. Acid will not break the peptide bond, but it'll open up proteins so that the access to the peptide bond is more readily available to pepsin. Okay, pepsin here, when it's in the presence of acid, autocatalytically activates the pepsin, and then more pepsin, a lot of pepsin. And here it's showing you that there's a protein, and pepsin makes smaller peptide fragments. There's no amino acids because pepsin is an endopeptidase. A long time ago, people used to put amino acids in the stomach and say, look what it does to acid secretion, motor effects, etc. And the problem is that probably in the stomach, you're not going to have very many amino acids. Also, the acidic contents of the stomach are within the pH optimum for pepsin. If you hear pepsin optimum pH is around 2 to 3, and it just compares it to the pancreatic enzyme here, trypsin, which is closer to 7, maybe almost 8. At any rate, everything is working. You have acid denaturing the protein and pepsin breaking the peptide bond. All right, this is a high school experiment, if you recall. 3 years ago, I probably presented the same slides. People put, I guess that would be a piece of chicken or something, in hydrochloric acid, pH 1.5 or whatever. And here they just added pepsin. And then here they added acid pepsin. And they came back, I don't know, a day later, 8 hours, 10 hours, I don't know. But the point being here is that in the presence of pepsin, you had some denaturing of this material. You can see the cloudy material here. But when acid was present along with peptide, it completely, solubilized the material, if you will. Okay, so in the presence of acid and pepsin, you can hydrolyze or digest this piece of chicken. So why doesn't the stomach digest itself? It's making acid and pepsin. And as I mentioned before, acid and pepsin will also kill other living things like bacteria. Okay, so this is actually one of the most important functions of gastric juice is to limit bacterial colonization. Um, and there's got to be a defensive mechanism that prevents your gastric mucosa from being digested, much like acid and pepsin did to this piece of meat. And that's what we're going to be looking at now, is the defensive forces. Sometimes when you're reading about this in textbooks, it'll be referred to as the gastrointestinal mucosal barrier. The word barrier is often used, and it is in a terms of barrier, both a chemical and physical barrier. Let's look at that. Usually, just like some GI pathophysiology you may run across, you can divide it into pre-epithelial, those events that are occurring in the lumen, okay? Epithelial, events directly attributed to the epithelial lining, okay? And subepithelial, and that's usually referring to the circulation, the capillaries, okay? You also have, we'll come to that later, immune system here, should some of these barriers break down. All right, let's look at the pre-epithelial, and here I have mucus, bicarb. And you can see in this slide here that these cells are generating bicarb, and at the surface of these cells you have pH 7, whereas in the gastric lumen you have a lower pH. Here they're saying pH 2, anywhere from 1.5 to 3 to some acidic pH. But you can see that there's a gradient being developed here with the epithelial, the surface of the epithelial cells not being exposed to acid. Okay, it protected. Well, let's take a look at that. We have the mucus here. We're showing a physical barrier, and I do this, and this is bacteria. Okay, and usually they're in the lumen, obviously, but they won't survive because, as I mentioned in the previous slide, acid and pepsin can kill bacteria. Okay, and also some enter into this upper portion of the mucus layer, okay, the outer loose layer. I'll define that in a minute. And usually they're confined to this area. But if mucus is presenting a kind of a physical barrier, because most microbes, let's say their average size is about a micron, and it's impeded by the mucin uh, gel, okay? This is what the mucin looks like. It's not just a fixed uh, mucus layer, if you will. It's a dynamic. That's what I've titled here. But it's subdivided just for discussion purposes into an outer layer, loose outer layer, and a firm or adjacent inner layer, which is very close to the epithelium. Terrible artist. I drew that horrible. Anyway, you have what's happening here is if the dynamic portion refers to that this inner layer is constantly replenishing this outer layer. Okay, and the outer layer is formed by pepsin in this case, or other proteases in the intestine, etc. They break some of the peptide bonds in glycoprotein, protein, and mucin, and so it allows it to spread and hydrate and become loose, if you will. And by loose, that is, it can be removed either by motor activity. If you recall, the stomach contracts 3 times a minute very strongly, and then you have retropulsion, etc. But the outer mucus layer, this loose mucus layer, is usually removed, emptied down to the small intestine. And I'm showing here the bacteria, okay, up here, acidic, and also here in the outer loose layer, the bacteria, 1 micron or so in size, diameter, if you will, they can get through and live here, but not very well because it's still acidic here, and I'll explain that in a minute. The pH neutral is closer to the epithelium here, but bacteria cannot penetrate very readily into this thick mucus layer, the inner or firm mucus layer. This layer is further not readily removed by motility or suction or something like that. But as you remove this layer, more mucin is secreted. So what's going on is, yes, you're losing the outer layer continuously, but you're also replenishing the mucus layer, okay, from by enhanced secretion of mucus. Okay, that's pretty straightforward, I think. Okay, well, let's take a look at I drew this line in here. This is the outer layer, inner layer, and they set up a gradient of pH. And it's usually, it's primarily in the inner layer because the outer layer is too loose to allow for a gradient. If you didn't have bicarb being generated, I'm not going to get into the transporters here, but you pick up bicarb and you put it into the thick, adherent, firm mucus layer, and it slows down diffusion in this layer substantially, such that the pH here at the surface is around 7, then it goes to 6.8, 6.5, 6.4, 6.3, and over here the acid and it becomes acidic. Okay, so in this inner layer, you allow for bicarb to neutralize some of the acid because it inhibits, to some extent, diffusion of these acid and bicarb. Okay. All right. If we look at the epithelium proper, you have tight junctions and you also have repair. I'll get to that in a minute. Tight junctions, that's shown here. Now, what are they? Tight junctions are these complexes that hold cells together. They're drawn. So occludins, zona occludins, and claudins are part of some of the proteins that make up these junctions. And this illustrates what happens with normal tight junctions. There's the bacteria or compounds made by bacteria or some dietary antigens, as they state here. They can't get through between these cells, okay? Now, if you don't have tight junctions or if they're disrupted, then these antigens can get through and they can activate immune cells in interstitium. If this activation is severe enough or strong enough, it can spill over into the systemic circulation and cause sepsis. So clearly, tight junctions are very important for this. And tight junctions, I think you'll if you read in the literature, they'll say that the stomach epithelium is a very tight epithelium, whereas the intestinal epithelium is loose, if you will. And all they're alluding to is that there's these tight junctions are more complex, and there's more of them in the stomach than there is in the small intestine. It's not that the small intestine lacks tight junctions; they're just less complex than those that are seen in the stomach. Okay, so that's the tight junctions. Now, the repair process, should these tight junctions break down or some cytotoxic material enter, and I like this injury, a little arrow here, lightning bolt or whatever, these cells are going to die, okay? Detachment, cell death. So what have you done? You lost these cells and you got a base, this is just the basement membrane, basically, and anything can get through. So what does the gastrointestinal tract do? It through a process called restitution, it makes a patch over the denuded area. Adjacent epithelial cells to this injury, they change shape. It's called epithelial mesenchymal transition or EMT, but they change shape and they begin to migrate, okay, over this denuded area, finally creating a patch, if you will. Now, restitution is very rapid. occurs within minutes to an hour, depending on whether you have 2 cells missing or 10. Then it'll take longer for this migratory. It's fairly rapid, though. And it does not involve cell replication or cell proliferation, if you will. And you note also that the cells here are not columnar, like you would see in the gastric mucosal epithelium, but they're flattened. Basically, it's a defensive maneuver to just seal the defect in the epithelial lining, but these cells are not really functional compared to these. Okay, they don't have the polarity, the transporters on one side, exchangers, etc., on one side versus the other. Okay, let's take a look at that. Restitution occurs within minutes. Yep, this is the small intestine. The same thing happens, in, just like a villi, obviously, and the same thing will happen in the stomach. It's just that this, because of the villi, you can more clearly see how this process works. Now you have damage. Something damaged epithelium. You lost the upper portion of the villi here, or in the stomach surface epithelium, and it's attracting your attention to these green adjacent cells adjacent to the defect. They change shape and migrate. Okay, this is restitution. It's basically covering the naked basement membrane to prevent excessive uh, movement of toxins or anything else that might be harmful into the interstitium. Okay, healing takes longer, and that requires proliferation in the crypt region. And you notice now functional cells, that is, polar columnar cells, start to occupy that area. And this occurs because, if you recall, epithelial turnover in the GI tract is rapid, 3 to 4 days. Every 3 to 4 days, some people say 5 days, you have new cells on the surface of the GI tract, okay? Which, is actually a good system to have considering the noxious material that's sometimes presented. Okay. Subepithelial, that will, as I mentioned before, is primarily the circulation, and we're dealing here with mucosal blood flow, okay? And that serves to wash out or neutralize aggressive factors. And usually it comes into play when the first two systems, the pre-epithelial and the epithelial, become dysfunctional, if you will. This is the gastric mucosa. Nice picture. You don't have to know this. Just suffice to know that there's the arterial supply and coming in the submucosal vein, submucosal artery, arterial supply, and it comes up into the gastric pits like this. Then they form these plexi, honeycomb-like appearance, and then they drain into veins, come down to the venous system. And remember, when acid secretion is going on, we mentioned that the gastric venous pH will actually go up. And this is showing you how the alkaline tide works. It's a blow up of this area, supposedly, and one can see here. All right, as you're making acid, okay, these parietal cells are making acid. That's in the wrong place, but that doesn't matter. You're making acid coming up here. Now, for every proton pumped into the lumen of the gastric gland, you have bicarb being exchanged for chloride, and to maintain pH neutrality in the cell, and it enters the circulation here. And because the way the circulation is designed, it goes up to the surface into these things, then comes down. This is referred to the bicarbonate flux, and this is the alkaline tide. It comes up here to the surface, and remember, we mentioned that these epithelial cells, mostly mucus, they put bicarb into the mucus coating to generate the bicarb gradient within the inner layer of the mucin. So it's a beautiful system because for if you make more acid, you make more bicarb to be transported up to neutralize the area very close to the epithelium in the inner thick layer, if you will. Okay? You don't have to know this. That's okay. So we've covered most everything that they do talk about in pathology: surface mucus, bicarbonate, mucosal blood flow, membrane. So you have to transport these bicarb, etc., to create the gradient. This is restitution. Now, prostaglandins, particularly the PGE tool, I mentioned that. Well, I'll mention it later. But the prostaglandins impact all defensive forces in a positive manner. Okay, let's take a look at that real quick. If you have increased prostaglandin, increased mucus, and bicarb secretion. So the mucus bicarbonate gradient is stimulated or promoted or enhanced by making more prostaglandins. And prostaglandins are continuously being made. Uh, I'll see if I covered that or not, but it doesn't matter. They come from arachidonic acid in the membrane. There's no specific cell type that one can allocate to secreting prostaglandins. Almost all cells can make them. Now, it also inhibits parietal cells, prostaglandins. That is, it reduces hydrochloric acid secretion, which is an aggressive factor. So it's promoting a defensive factor, inhibiting an aggressive factor. The other thing it does, it maintains mucosal blood flow, which is good, as we discussed earlier. Also enhances epithelial restitution, which is good. So it's prostaglandins are impacting all of the defensive forces that we talked about uh, to maintain the gastric barrier, if you will. Okay? All right. Now, this is extension of the slide we've been dealing with, and here they're showing you injury. And the way you're going to get any pathology in the stomach is either through increasing your damaging forces and or decreasing your defensive forces. Okay, and we'll cover the two main ones here. They have H. pylori and NSAIDs. I think you know what non-steroidal anti-inflammatory drugs. And aspirin is also considered an NSAID. I don't know why they separated here. Alcohol is not an issue, and usually in Saudi Arabia anyway. Um, so you're going to have increased damage or impaired mention that, and you get ulcers. Okay, so let's cover H. pylori and NSAIDs. Here's the NSAIDs, and what do they do? NSAIDs inhibit cyclooxygenase. That's COX. Cyclooxygenase 1 is a constitutive form, whereas cyclooxygenase 2 is supposedly the inducible and pro-inflammatory one. The constituent form is usually referred to as the housekeeping prostaglandin because they're always making it, the prostaglandin, as I mentioned. At any rate, almost all your NSAIDs inhibit COX-1 and COX-2 to different degrees. I'll come to this later. It's not that important, but it inhibits basically prostaglandin production. Don't worry about thromboxane. But PGE2 is a major player in the gastric mucosa, as well as prostacyclin, which is PGI2, same thing, prostacyclin. Let's take a look at this. Remember, this is a slide from before about prostaglandins in here. If you decrease PGE2, you're decreasing mucus bicarb. What are you doing? You're decreasing your defense against the acid and pepsin, okay? And why do I talk about acid and pepsin? Well, well, now that I have it here, I might as well talk about it. You have acid coming up here in pepsin. And if you think about this slide here, if you have pepsin here and you decrease your gastric bicarbonate in the inner mucus layer, you're also allowing pepsin to attack the cell. That's why it's called peptic ulcer disease. And the reason for that is because pepsin here is active at a low pH, but as pepsin approaches the lumen, where the the epithelium, where the pH is high, then its optimal activity is reduced. It's not operating anymore. It's ineffective. at a neutral pH, as we showed you before, I think, somewhere. Anyway, all right. So you inhibit mucus bicarb production, so you have acid peptic injury. Uh, well, you're reducing prostaglandin E2. You're going to increase acid secretion. Remember, ordinarily, prostaglandins inhibit acid secretion. Now you're increasing acid secretion. Also, the same thing, decrease in prostacyclin reduces mucosal blood flow. Mostly PGE2, prostacyclin, mostly, they're all vasodilators, most of the prostaglandins. Okay, so they maintain mucosal blood flow. You don't have to know this. I mentioned this, that prostaglandins are generated from arachidonic acid. You probably had all this in pharmacology or biochem. Let's summarize non-steroidal anti-inflammatory drugs. Okay, all your defensive forces are down. So they affect the pre-epithelial, epithelial, and subepithelial defensive force. You got decreased mucus, decreased bicarb, decreased restitution should the epithelium break down. You have a decrease in mucosal blood flow, which inhibits all kinds of functions, obviously, and you have an increase in acid secretion. All right, so so it's decreasing your defensive forces, increasing the damaging forces. Now, it also has a direct cytotoxic effect to increase epithelial cell injury. Uh, I think we cover that here? Yes, okay. So NSAID, we talked about this. Here they're just emphasizing COX-1, or else I blocked out the other ones. But you have decreased in prostaglandin E2 synthesis here, they're showing you. And as I mentioned before, it's a vasodilator. Prostacyclin is more potent, but don't worry about that. You have prostaglandin, decreased mucus, decreased bicarb, decreased blood flow, increased acid, okay? Now, that mucosal damage, epithelial injury, now you have acid and pepsin work to induce damage. You also have vasoconstriction because prostaglandins usually maintain blood flow. And also you have topical irritation, direct damage. NSAIDs interact with tight junctions and disturb them, and they also damage epithelial cell mitochondria. At any rate, everything here NSAIDs causes. Well, you can't avoid NSAIDs because they're pain relievers, okay, non-steroidal anti-inflammatory drugs. I'll come back to this in a minute or in a few minutes. The other common cause of gastric ulcers or peptic ulcer disease is Helicobacter pylori. And this slide is a little old, but still 50% of the human population is believed to be infected with H. pylori. But obviously, here in Saudi Arabia or other developed countries, there's going to be much less of the population is going to be infected, whereas in other areas where either ravaged by war or poor hygiene, etc., more prevalence of H. pylori infection. And some people, based on genetic analyses, suggest that H. pylori has been associated with humans for up to 60,000 years, if you want to believe that. I don't know how they get these numbers, but it's been around for a long time. So people are wondering, well, it must be doing something good, else we would have gotten rid of it by now, just by a natural selection, if you will. Now, 20% of H. pylori people will have ulcers. Okay, so not everyone who's infected by H. pylori will have an ulcer, but almost all the people that have ulcers will also be positive for H. pylori. You could think about that one for a while, but you'll see that it makes sense. But at any rate, H. pylori, this nice little picture here. The person who came up with this, he actually drank H. pylori. He deserved a Nobel Prize for that, and that's what he had. Anyway, here is a nice figure. Obviously, I didn't draw it, but here you have the gastric lumen, and what they show you here is outer mucus layer and the inner mucus layer in the actinic gland, in the pyloric gland area. This will be the fundic region, antrum. So it's, okay. So here they're showing you bacteria, and they're showing you it's in the, mostly in the outer mucus layer, as I mentioned. And they're pointing to H. pylori here, this flagella. Okay, so that's H. pylori in here, and other kinds of bacteria here. Now, what does flagella do? It allows them to escape the acid pepsin um, degradative process, if you will. And just like any other bacteria, H. pylori can be killed by acid and pepsin. And I think some estimates are that maybe only 1% or less of the inoculated H. pylori actually survive to colonize the stomach. So 99% of all the H. pylori that comes in gets killed, but 1%, all you need. Okay, so here you have H. pylori can have flagella, and they can enter the mucus layer. Okay, but that's not enough. They have another thing working for them as well, and that's something called spatial orientation. They naturally tend to move to a neutral pH. If you put H. pylori in the middle of a slide somewhere, put acid on one side and neutral pH on the other side, they'll always move, or most of them will move towards the neutral pH away from the acid. So both of these, the flagella and their ability to orientate themselves toward the neutral pH, allows them to occupy the inner mucus layer, and that's what presents us with the problem with H. pylori gastritis, if you will. And here it's showing, you see another thing here? Here's your H. pylori colonizing the inner mucus layer, okay, of the antral region, but only staying here. They don't go into the pit here in the fundic region. And the major reason for that is outlined here in red, is that your parietal cells are located in the fundus, okay? They make acid, and H. pylori is not going to move down there, okay? Whereas in the antrum, it will. And this also some people use as an explanation for why the initial infection with H. pylori is usually in the antrum. The other thing the H. pylori has is urease, which allows it to create an ammonia cloud around it. And ammonia being basic helps protect it against acid, if you will, acid pepsin as well, because remember, pepsin requires a pH optimum of 1. And this is actually, clinicians came up with a urea breath test, which can be done in an office of a general practitioner, and basically you ingest carbon labeled urea, and urea only has one carbon. Okay, you have an ammonia here, one, another ammonia, and urease will generate carbon dioxide and ammonia from urea. All right, so you're ingesting carbon laden urea. The urease will form the only carbon. It's labeled now, ends up in the blood, and you can measure it in the breath. Okay. So that here a normal person, or not a normal, a person who's not infected with H. pylori, that's what there will be no radio-labeled carbon because the urea is not broken down by urease. Okay, all right. And this is the breath test. Very straightforward. You have your patient breathe into it and then send off for analysis. This is a summary. H. pylori. I didn't write this yet. We use flagellas to penetrate towards, or did I? I don't know. Towards the mucus layer down inside here. Okay, using urease. It's very popular to talk about urease and the ammonia cloud here, which neutralizes gastric acid. But I would propose that the flagella and its ability to orientate towards a neutral pH is probably more important. At any rate, it escapes. It tries to escape from the detrimental effects of acid pepsin, colonization, and eventually destruction of the mucus. They have mucinase, and they secrete toxins, and here you have inflammatory cells coming in, like neutrophils. So it causes inflammation. Okay, this is an old slide, 10 years old, but it still applies today. You have people who are NSAID users and controls with non-NSAID users, okay? And then you have H. pylori, they measured H. pylori, urease breath test, if you will. So if you don't have H. pylori and you're not using NSAIDs, the chances of you having ulcers is fairly remote. If you're H. pylori positive, but not using NSAIDs, then about 20% will have gastric or peptic ulcers, if you will. And here you have H. pylori negative, but they're using NSAIDs, again, 20%. But if they're both NSAID users and are colonized by H. pylori, almost 50% chance of having peptic ulcer disease. So people are now interested in eliminating the H. pylori for one thing, and then trying to curb the use of NSAIDs. That's a difficult thing to do because NSAIDs are very potent anti-inflammatory agents, and they help with pain, like aspirin, normally used for headaches, etc., or acetaminophen against pain. Generally, you can buy them over the counter usually. And drug companies are very much interested in finding formulations that will inhibit COX and not cause injury. And one of the original approaches was to say, okay, you're having gastric ulcers, but the patient has to be on pain relievers for, let's say, rheumatoid arthritis or something. But how do you avoid the gastric ulcer? Well, I'll show you that in a minute here. This just shows you how many uh, different NSAIDs there are and their relative different selectivity. Some companies try to get formulations that inhibit the COX-2, the pro-inflammatory cyclooxygenase, more than the COX-1, which is the housekeeper. And on this spectrum, you have more, they inhibit more the housekeeping function. With aspirin. Here's aspirin, for example. With ibuprofen is here. I think you're familiar with some of these. Anyway, some have run into problems with intestinal injury or cardiovascular issues, and I'm not going to get into that. But there's probably even more now formulations appearing. What's really helped this area, as I mentioned, people are looking at different NSAIDs, trying to find one that alleviates the pain that doesn't cause injury. And one of the earliest approaches that I mentioned was to uh, see if you could get this NSAID not to become dissolved in the stomach, but survive the stomach acid and pH and end up in the small intestine, right? And well, they did this, and it was amazing. Gastric ulcers did not occur in these people now with these formulations of NSAIDs, okay? But they didn't know it until this kind of techniques because endoscopy couldn't get down to the small intestine. Basically, what they were doing is just moving the injury downstream. So what you had is, yes, slow release tablets or specially coated tablets would make it to the stomach, to the small intestine, and the stomach's just fine. But now the small intestine's injured, okay, due to the direct toxic effects. And this was readily established by using these capsule endoscopy, if you will. And basically, it's a small camera with a lens, illuminating lens camera. It's a small camera in a pill that or pill cam. Okay, so it's called capsule endoscopy. And this is some of the injuries, and this is in the intestine now in response to NSAID, and everywhere from irritation to mucosal ulceration, active bleeding, and stricture formation You don't have to know this, but just to show you, all they did was they're moving the NSAIDs down. This is a last slide, and all it illustrates that some of these NSAIDs can actually enter the enterohepatic circulation. They get conjugated with glucuronide, which can increase or decrease their toxicity. And here it says plus bile. Some investigators have shown that the bile can actually potentiate the toxic effects of some of these NSAIDs. Okay, well, that's all I have. And good luck. Did I go over time? No, I'm good.
Thank you,
If there's
Dr.
any questions, I don't mind dealing with them. But if not, have a good day then. Are there?
Thank you, Doctor. One of the students was asking if you can kindly share your slides or upload them to Moodle.
Do what now?
If you can upload the lecture on Moodle.
Oh, you want me to post it on Moodle?
Yeah, please.
No problem. I will do that. I don't know if I have access. I'll find out from Dr. Woban. Or I think he's lecturing next. So ask him, remind him, and he'll give me access. Or I'll send it to
Okay,
him. He'll post it.
if you would like the email I sent yesterday for the Zoom
Yeah.
link, you can reply to it with the lecture and I can forward it to the students.
Okay. I will try to do that. If it moves
Yeah.
the whole, okay, I will do that. All right.
Okay.
All right. Have
Thank
a
you
good
so much.
day. Yep.
Have a good day.
Bye.